The mechanism of translation could be divided into distinct stages. For example, the first stage is called as initiation, which is followed by elongation and at the end by termination of translation. One by one, we will discuss these stages. For the translation process to occur, a template, which is nothing but the mRNA, which has the information for various codons, is required. This mRNA will have the information for various codons of amino acids that are going to form the polypeptide. Here we have the start codon, that is AUG on the mRNA molecule, remaining codons for other amino acids, and at the end we have a stop codon, that is the termination codon. This template will be copied during the process of translation. This mRNA is going to attach to the ribosomal subunits, the smaller and the larger one. Together they form the 70th subunit of prokaryotic ribosome. The codons for various amino acids that are represented on the mRNA, these are brought to the site of protein synthesis, that is the ribosome, by a tRNA molecule. This tRNA molecule will have an attached amino acid. So this is the first tRNA molecule which have the first amino acid attached to it. So in response to the start codon, the first amino acid, that is N-formylmethionine, will be brought up by this particular tRNA molecule. So during the initiation step, when the AUG codon it is present at the P site of the ribosome, the first amino acid is brought to the ribosome. And this first amino acid within the prokaryotes is nothing but the N-formylmethionine. During the elongation step, one by one, the other tRNA molecules are brought and they are added to the A site of the ribosome. The protein chain is formed by attachment of one amino acid to the another. So the first amino acid that was brought by the tRNA molecule which was present at the P site is now attached to the second amino acid which is present at the A site. One by one, the tRNA molecules will bring the amino acids and these are attached by peptide bond to form a polypeptide chain. This is followed at the end by attachment of release factors when the termination codon comes into the reading frame. So here we have the termination codon in the reading frame. The release factors will come and attach themselves at the A site. In response to that, the components of the translation, that is mRNA, polypeptide chain which has been synthesized, the smaller and larger subunit of ribosomes, they all separate from one another. And the cycle of translation comes to a halt. The translation or protein synthesis mechanism within the prokaryotes, it is nothing but the decoding of mRNA. The codons that are present on an mRNA molecule, they provide the information of the type of amino acids that are to be brought at the site of protein synthesis. These amino acids, these are linked together by peptide bond to form a protein or a polypeptide. In order to carry out the translation mechanism, the components that are required include mRNA. Now we know that within the prokaryotic organisms, a polycystonic mRNA is present. So first requirement would be polycystonic mRNA, which represents the information for more than one protein. This mRNA, it is acting as a template for the translation. This mRNA within the prokaryotic organism, it has a very short lifetime. Second component required is a tRNA molecule, transfer RNA molecule, whose function is to transport the amino acid to the site of protein synthesis. During translation, the amino acids, they are linked together by peptide bond to form the functional protein. So amino acids, they are also an important requirement. All this mechanism of translation is carried out at the ribosome. So ribosomes, they represent the site of protein synthesis. Ribosomes are formed by a combination of ribosomal RNA plus ribosomal proteins. Apart from these components, the enzyme amino acid tRNA synthetase is required. Enzyme amino acid tRNA synthetase, it is involved in attachment of amino acids to tRNA molecule. With these important components, some other important resources that participate in the translation mechanism include energy sources. The energy sources such as GTP, elongation factors, and the release factors. The first step in the translation mechanism is activation of amino acid, which is brought about by the enzyme amino acid tRNA synthetase. Activation of amino acid is carried out with the help of an energy source that is called as ATP. Amino acid will first interact with the ATP molecule to form amino acid AMP complex 1 pyrophosphate. One molecule of pyrophosphate is released during that step. The amino acid from the amino acid AMP complex is then transferred to a specific tRNA molecule and AMP, that is adenosine monophosphate, is released. Both the steps, that is interaction of amino acid with ATP to yield amino acid AMP complex and amino acid from the amino acid AMP, its attachment to a specific tRNA molecule and release of AMP is brought about by the enzyme that is called as amino acid tRNA synthetase. This activation of amino acids is carried out within the cytoplasm by the enzymes that are called as amino acid tRNA synthetases. In this manner, all 20 amino acids will be activated first and then attached to the 3' end of the specific tRNA molecule in presence of ATP and magnesium. The initiator tRNA, it is formed by addition of N-formylated methionine. It will add the first amino acid N-formyl methionine during the protein chain synthesis. All 20 amino acids, these are activated by the same mechanism and then bound to the specific tRNA molecules forming amino acid tRNA. The charging of tRNA molecule, that is attachment of amino acid to the tRNA, is carried out by amino acid tRNA synthetase enzyme. So here we have this amino acid tRNA synthetase enzyme. To this enzyme, the amino acid and an energy source such as adenosine triphosphate, that is ATP, will come and attach first. This particular amino acid and ATP will bind to the enzyme. The adenosine monophosphate from the ATP, it is covalently bound to this amino acid and the 2-phosphate, that is the pyrophosphates, these are removed from this complex. Now here we have only the amino acid along with AMP complex. The correct tRNA, it should bind to this particular enzyme. A tRNA molecule here will come and interact with the amino acid which is present at the amino acid tRNA synthetase. When this tRNA gets attached to this amino acid, the AMP adenosine monophosphate, it is released from this enzyme. Now we can say that the tRNA molecule is charged. That is, an amino acid is attached to that tRNA molecule. Such type of charged tRNA is then released from the enzyme, that is amino acid tRNA synthetase. This amino acid is attached to a particular terminus of the tRNA molecule. This is attached at the 3' hydroxyl terminus of the tRNA molecule by an ester bond. The same mechanism of charging of tRNA molecules, which is followed by activation of amino acids, is being shown within this slide. This is the amino acid that is going to be attached to the tRNA molecule. An amino acid, along with energy source such as ATP, bind to the enzyme, that is amino acid tRNA synthetase. The inorganic pyrophosphate is removed from this ATP. After removal of inorganic pyrophosphate, AMP will, adenosine monophosphate, will remain here. So enzyme, that is the amino acid tRNA synthetase, it gets attached with adenosine monophosphate, that is AMP, and the amino acid. So this complex is comprised of enzyme plus adenosine monophosphate and amino acid. All these three components are linked together. So enzyme, amino acid and AMP. This is nothing but the activated amino acid. This is also referred by another term, amino acid AMP enzyme complex. From this complex, after interaction of a tRNA molecule with this amino acid AMP enzyme complex, the amino acid is attached to the tRNA molecule. So initially, a tRNA molecule will interact with this amino acid AMP complex. This tRNA molecule, to that, this amino acid will be attached. And during that mechanism, the adenosine monophosphate within the complex is released along with the enzyme. So these two components will be separated. So AMP and enzyme, they will get separated and only the amino acid will be attached to the tRNA molecule. Such type of tRNA with an attached amino acid is called as amino acid tRNA. A cell will have a collection of tRNA molecules with attached amino acid. Every tRNA molecule will have a particular type of amino acid attached to it. Their function is to bring that attached amino acid to the site of protein synthesis during translation. In the initiation of translation, the first step is that the initiation factor number 3, that is IF3, it is binding the smaller subunit, that is 30S ribosomal subunit. The mRNA, which is a template for the translation, it binds to the 30S ribosomal subunit. Its binding is such that AUG codon, it lies at the P site, that is the peptidyl site present on the 30S ribosome, whereas the second codon, it lies at the amino acyl or A site. The initiator tRNA, it carries the formilated methionine by initiation factor number 2. With the hydrolysis of GTP, this formyl methionine tRNA, it is placed at the P site on the smaller subunit of ribosome, that is the 30th subunit of ribosome. The initiation factor number 1, its function is actually to prevent the binding of formyl methionine tRNA in the A site on the 30th ribosomal subunit. So till initiation factor number 1 is attached to the 30th subunit, the formylmethionine tRNA cannot be placed at the A site. It is actually blocking the A site. By blocking, it is preventing the attachment of formylmethionine tRNA in the A site. So because of this reason, then the formylmethionine tRNA it is placed at the P site. During the initiation of translation, the binding of the 30S subunit to the mRNA molecule, that is the template for the translation, a sequence that is present on mRNA that is called as Shine-Dalgarno sequence. It plays a very important role. This particular sequence, it is actually guiding for the correct positioning of the AUG codon on the P site of the 30S ribosome. So binding takes place between the SD sequence that is present on mRNA and the 16S rRNA that is present on the 30S ribosome. SD sequence is rich in purine and the 16S rRNA that is present on the 30S ribosome, it is rich in pyrimidines. The two sequences are complementary to one another. Due to the complementarity of bases between the SD sequence of the mRNA and the 16S rRNA that is present on the 30S ribosome, the binding between the two takes place. This helps in the correct positioning of the first codon, that is the start codon AUG, at the P site, that is the peptidyl site on the 30S ribosome. After the binding of this particular mRNA to the 30S or smaller subunit of ribosome, this is followed by binding of another tRNA molecule at the P site. That tRNA molecule is called as initiator tRNA or also called as FMET tRNA. FMET is nothing but formylmethionine tRNA. Formylmethionine tRNA. This particular tRNA, it is brought or it is loaded onto the P site or the peptidyl site on the 30th subunit with the help of initiation factor number 2. Once the addition of this formylmethionine tRNA is carried out at the P site, then the three initiation factors, initiation factor number 3, number 2 and number 1, they are released. The function of the initiation factor number 1 is to block the A site on the 30th subunit so that the initiator tRNA does not load the formylmethionine tRNA at the A site. Initiation factor number 2, it is carrying out the function of loading of this initiator tRNA with formylmethionine at the P site. The function of the initiation factor number 3 is quite unique. When this protein is attached to the 30th subunit of the ribosome, it prevents the binding of the larger subunit to the 30th initiation complex. So till all the three, number 1, number 2 and number 3, initiation factors are attached to the 30th subunit, the 50th subunit cannot attach. Once the 30th initiation complex is established by addition of formylmethionine tRNA on the P site, these initiation factors are released from that initiation complex. After release of these initiation factors, the larger subunit, that is the 50S ribosomal subunit, then binds to the smaller, that is the 30th subunit, forming a 70S initiation complex or a 70S ribosomal complex. Once the larger subunit it is joining the smaller subunit, then all the three important sites they are exhibited on that 70S ribosome. It will have the A site, that is amino acid site, represented on it, the middle P or peptidyl site, and E, that is the exit site, that is located in the 50S ribosomal subunit. This slide represents the basis for attachment of 30S or smaller subunit of ribosome to the mRNA molecule. Attachment of an mRNA molecule to the smaller subunit of ribosome, it is due to the two sequences that are present on the mRNA and on the ribosome. On the mRNA molecule, there is an SD sequence, Shine-Dalgarno sequence, which has complementary nucleotides to the 16S rRNA that is present in the 30S subunit of the ribosome. Now we know that ribosome structure, it is constructed by a combination of rRNA molecule with the ribosomal proteins. So the rRNA molecule that is present in the smaller subunit of ribosome, that is the 30th subunit of ribosome, it is 16S rRNA. This 16S rRNA has a sequence which is complementary to this SD sequence. It is with the help of this complementarity of nucleotides between the SD sequence and the 16S rRNA, the mRNA it is binding to the smaller subunit of ribosome with its 16S rRNA component. First step in the initiation of translation is that the initiation factors, initiation factor number 1 and initiation factor number 3, they bind to the 30S ribosomal subunit, which is the smaller ribosomal subunit. This complex of initiation factor number 1, 3 along with the 30S subunit then bind to an mRNA molecule, which is a template for the translation. This mRNA molecule has a particular region which is called as Shine-Dalgarno sequence. This Shine-Dalgarno sequence, it is also called as SD sequence. This sequence is also referred by another term that is called as ribosome binding site. It is this site which helps in binding of mRNA to the ribosome. This ribosome also has a particular section within it that is called as 16S rRNA. This 16S rRNA that is present in the 30th subunit of ribosome, it is rich in pyrimidine. Shine-Dalgarno sequence is rich in purine. So there is a complementary sequence between the Shine-Dalgarno sequence that is rich in purine and the 16S rRNA sequence that is present within the 30th subunit of ribosome. The complementarity of these two sequences helps in binding of 30th subunit to the mRNA molecule. So here you can see the 30th subunit with its 16S rRNA sequence is binding to the mRNA molecule. So mRNA molecule is shown in green color within this particular image. After binding of this mRNA to the 30th subunit, then the next step is the initiator tRNA. Initiator tRNA is a first tRNA molecule which is bringing the formylmethionine. So this is an initiator tRNA molecule. The secondary structure of that tRNA molecule is shown in the image, and a formylmethionine amino acid is bound to the 3' end of that initiator tRNA. Such type of tRNA molecule is called as F-met initiator tRNA. F-met is nothing but formylmethionine initiator tRNA. Such type of tRNA molecule, which is called as initiator tRNA with a bound formylmethionine, it is then coming and binding to the 30S ribosomal subunit mRNA complex. Now this particular tRNA molecule, initiator tRNA molecule, is brought with the help of initiation factor number 2. So here we have this initiation factor number 2, and with the help of this initiation factor number 2, which is a GTPase protein, this particular tRNA, initiator tRNA with a bound formylmethionine, is brought to the 30S ribosomal subunit mRNA complex. So GTP hydrolysis, the energy that is used for the addition of this particular tRNA molecule to this complex is GTP. GTP will undergo the hydrolysis to form GDP plus inorganic pyrophosphate. So GTP hydrolysis energy expenditure is required for addition of initiator tRNA at the P site of this particular ribosome. So here we have the E site, P site and the A site on this particular ribosome. Initiator tRNA is always added to the middle P or peptidyl site. The rest of the two sites, amino acid site and the exit site, they are free. After the addition of the formylmethionine initiator tRNA with the bound formylmethionine group at the P site of the 30S ribosome mRNA initiation complex, this is the structure that is created. Here we have the smaller subunit of ribosome shown in blue color. It is attached to the mRNA molecule by the 16S rRNA sequence that is present within the ribosome and the SD sequence that is present on the mRNA. There is a bound initiation factor number 3, bound initiation factor number 1 and a tRNA that is brought about with the help of initiation factor number 2. Now if you see the positioning of this first initiator tRNA with a bound formyl methionine, this particular tRNA is placed at the P site of the ribosome, and at the P site there is the first codon or the start codon of that particular mRNA molecule. So here we have the start codon, that is AUG. A complementary sequence exists on the tRNA molecule which is bringing the formyl methionine. Here the complementarity can be easily observed. Uracil is paired with adenine, adenine is paired with uracil, cytosine is paired with guanine. So the first tRNA molecule, which has a anticodon sequence complementary to the start codon, will bring the formyl methionine to the site of 30S initiation complex. Hydrolysis of GTP is required for loading of this tRNA molecule by initiation factor number 2. Now, right now we know the function of initiation factor number 2, that is, it is used for loading of the initiator tRNA by hydrolysis of GTP at the P site. Now the A site and the E site are free. Actually, the A site or the amino acid site that is present on the ribosome is not used for loading of this initiator tRNA. The reason behind that is initiation factor number 1, it is bound to this 30th subunit of ribosome in such a manner that it will not allow the binding of this initiator tRNA at this position. So this site, A site amino acid site, is blocked by initiation factor number 1. So function of initiation factor number 1 is to block the amino acid site so that the initiator tRNA cannot be added at this particular position. It can only be added at the P or peptidyl site. The initiation factor number 3, it also serves an important function in the 30S initiation complex. When this initiation factor is attached to the smaller subunit of the ribosome, it will not allow the attachment of larger subunit to this smaller subunit. So it does not allow the attachment of larger subunit of ribosome to this 30S initiation complex. So till this particular initiation factor number 3 is there, no attachment of 50S subunit is possible to this 30S initiation complex. So this 30S initiation complex, it has initiation factor number 1 which is blocking the amino acid site, initiation factor number 3 which is preventing the attachment of larger subunit to this smaller subunit, and it has initiation factor number 2 which has carried out the loading of the initiator tRNA at the P site of the ribosome. It has brought the formyl methionine to the site of 30S initiation complex. So this is the 30S initiation complex. After the formation of 30S initiation complex, the next step is the initiation factor number 1, number 2 and number 3. They get separated from the smaller subunit of the ribosome along with the GDP and inorganic phosphate. This 50S ribosomal subunit, which is also called as the larger ribosomal subunit, then comes and attaches to the 30S initiation complex, ultimately forming a 70S initiation complex. So here we have the smaller subunit of the ribosome with a bound mRNA molecule, attached initiator tRNA with formyl methionine at the P site of the ribosome, whereas this is the larger subunit of ribosome that has attached to the smaller subunit after removal of initiation factor number 1, number 2, number 3 with the hydrolysis of GTP to GDP and inorganic phosphate. So in this manner, the whole ribosome assembly of 70S initiation complex, it is established. Now we have this A site at this position which is free. P site has initiator tRNA with first amino acid in the sequence which is formyl methionine, and the E site is also free. So A site and E site both are free, and P is occupied by the initiator tRNA along with the 70S ribosome, smaller subunit and larger subunit, they are together. So we can call this as a 70S ribosome which is attached to the template for translation, that is mRNA. After the initiation of translation, the next step is elongation. During this stage, amino acids are added one by one to form a polypeptide chain. Addition of each amino acid occurs via a series of steps. This process is complex. In bacteria, approximately 15 to 18 amino acids are added per second. Within the eukaryotes, this process is slightly slow. Addition of 6 amino acid takes place per second. Next step is binding of amino acid tRNA to the A site on the ribosome. The second tRNA molecule, it will carry the next amino acid and will add this tRNA at the A site of the ribosome. Once the initiator tRNA with the first attached amino acid is loaded on the P site, in the next step, another amino acid tRNA with its attached amino acid. This amino acid is specified by the second codon on the mRNA molecule. So, amino acid tRNA, which has an anticodon sequence complementary to the second codon on the mRNA molecule, will bring the respective amino acid and load it at the A site on the ribosome. This binding of amino acid tRNA at the A site, it is carried out with the help of elongation factor, which is called as elongation factor Tu. E represents elongation and F represents the factor. Elongation factor Tu, it is utilizing energy derived from GTP. So binding of amino acid tRNA to the A site requires energy source, that is guanosine triphosphate GTP. After its binding to the A site, the GTP is hydrolyzed. GTP hydrolysis converts the elongation factor Tu-GTP to elongation factor Tu-GDP. This GDP is released from the ribosomal complex. Elongation factor Tu-GDP, it then enters into elongation factor Ts cycle. Once the amino acid tRNA with a bound amino acid is brought and added to the A site on the ribosome by elongation factor Tu with the hydrolysis of attached GTP, the amino acid that is present on the tRNA molecule at the P site, that is the formyl methionine, is transferred to the tRNA that is located at the A site. These two amino acids, that is the formyl methionine and the next amino acid that is brought about and loaded at the A site, these two, they join together to establish a peptide bond. Formation of peptide bond is catalyzed by an enzyme that is called as peptidyl transferase. Now, the tRNA that is present at the P site, it becomes uncharged. The amino acid from that tRNA has been transferred, or it is joined by peptide bond to amino acid that is present at the A site. In the next step, that is translation elongation, the codons beyond the start codon, they are read one by one by various tRNA molecules. These tRNA molecules that are called as amino acid tRNA with their attached amino acids are brought and loaded at the A site on this ribosome. The loading of this particular tRNA, amino acid tRNA with an attached amino acid, is carried out with the help of elongation factor Tu with a bound GTP. Hydrolysis of GTP takes place during the loading of such amino acid tRNAs at the A site. Once this introduction is carried out at the A site, then the amino acid or the amino acid chain that is present with the tRNA at the P site is then linked to the amino acid that is brought about by the amino acid tRNA. Establishment of peptide bond takes place. So the whole polypeptide chain, or even if there is a single amino acid at this location, this is transferred to the amino acid, or this is linked to the amino acid that is present at the tRNA present on A site of the ribosome. This peptide bond formation, it is catalyzed by peptidyl transferase activity. Peptidyl transferase activity, it is present on the larger subunit of ribosome, and it is actually carried out by 23S rRNA molecule, which is a component of larger subunit. So one by one, the amino acids that are brought to this site of ribosome, they are linked together by peptide bonds. Now after separation of the amino acid from the tRNA at the P site and loading it on the linking it on the amino acid present at the A site, this tRNA becomes free. It does not have the attached chain or attached amino acid with it. The translocation movement is carried out further. During the translocation, the ribosome moves along the mRNA molecule by one codon. Due to this, now the free tRNA molecule, which does not have any attached amino acid, will move towards the exit site, E site. The amino acid, the tRNA molecule which is present at the A site with the attached polypeptide chain, will move towards the P site, and the A site becomes free. Again, one more amino acid tRNA will read the codon that is present at the amino acid site, bring the attached amino acid with the help of elongation factor Tu with a bound GTP, introduce that tRNA at this position. Then the polypeptide chain of the tRNA that is present at the P site, it will be linked by peptide bond to the amino acid that is brought about and placed at the amino acid site. So one by one, the amino acids will be brought about by the amino acid tRNAs, placed at the A site. The polypeptide chain which is linked to the tRNA at the P site is then removed. This linkage between the tRNA and polypeptide chain is degraded, and the whole chain is then linked by peptide bond to the incoming amino acid brought about at the A site. Again, the translocation will take place. Now translocation is carried out. The movement of the ribosome along the mRNA molecule by one codon, it is carried out with the help of elongation factor Tg. After the peptide bond formation takes place between the first and second amino acid, the ribosome moves one codon ahead along the 5' to 3' direction on mRNA molecule. Because of this reason, the dipeptide tRNA, now it will be shifted to the P site, and the next codon in sequence, it appears on the A site. The uncharged tRNA molecule from the ribosome that is present at the E site now will enter into the cytoplasm. It will exit from the ribosome and enter into the cytoplasm. This ribosomal translocation requires a factor that is called as elongation factor G, which is bound to GTP. This is also referred by another term that is called as translocase enzyme. It changes the 3D structure of the ribosome to catalyze its 5' to 3' movement by one codon. Due to translocation, now the codon that is present at the A site, it is recognized by another amino acid tRNA, which has an anticodon sequence complementary to this codon. It brings the respective amino acid to the site of protein synthesis and load it at the A site on the ribosome. The dipeptide, which is present on the tRNA at the P site, is then transferred to the amino acid that is present at the A site. It forms a tripeptide amino acid chain. During the translocation, the amino acid chain which was initially present on the tRNA that is located at the P site, it is transferred to the amino acid which is present on the tRNA at the A site. Now this particular tRNA, it has become uncharged. It does not have any amino acid linked to it. Due to translocation, this tRNA molecule is going to shift to the E site The tRNA, which has a chain of amino acid, a short polypeptide here, is going to shift to the P site and the A site is going to become free. The ribosome, it is actually moving by one codon during the translocation, and this helps in changing the position of the tRNA molecules on the ribosome. This translocation is brought about by elongation factor G, which is bound with GTP. Its translocase activity, it is an enzyme requiring, it is an energy requiring activity. This translocase enzyme is then helping in movement of ribosome by one codon. So once the shifting takes place, shifting of ribosome by one codon takes place, then you can see here the A site is free. The tRNA molecule, which had a long peptide chain and was present at the A site here, has now shifted to the P site. The uncharged tRNA molecule, which was present at the P site, is now present at the E site. E site is nothing but the exit site. Now the polypeptide chain, it is with the tRNA molecule which is present at the peptidyl site. So this is amino acid number 1, 2, 3 and 4. One by one they have been linked by peptide bond by peptidyl transferase activity. The uncharged tRNA, it is released from the E site into the cytoplasm. This process goes on repeatedly, in which one by one the amino acids are brought by tRNA molecules by reading the codon that is present at the A site. The newly brought amino acid to that the polypeptide chain is linked. This tRNA will become uncharged. Ribosome translocation will move the ribosome along the mRNA in 5' to 3' direction by one codon. So one codon movement towards the 3' end takes place. Now the uncharged tRNA will shift to the E site. The tRNA molecule which has received the polypeptide chain will come and shift itself to the P site, and the A site will again be free for incoming amino acid tRNA, which will bring a new amino acid. So this process will go on repeatedly during the elongation again and again until it reaches a stop codon. During the elongation phase of translation, every time when a new amino acid is brought to the site of protein synthesis, peptide bond formation between that amino acid and pre-synthesized polypeptide chain takes place by peptidyl transferase activity. This type of elongation of polypeptide chain, it is going to continue until a stop codon appears at the A site on the ribosome. If the stop codon it appears on the A site, then it is not recognized by any tRNA molecule which is carrying the amino acid. This is because the stop codon do not have anticodons present on a tRNA molecule. Stop codons, instead of being recognized by tRNA molecules, they are recognized by another protein which is called as release factor. Within the prokaryotic organisms, there are three types of release factors: release factor number 1, 2 and 3. Function of these release factor is to carry out the hydrolysis. Hydrolysis by the release factors, it causes release of all components of translation, that is the 30S, that is a smaller subunit, 50S, that is the larger subunit, the template, that is the mRNA, and the newly synthesized polypeptide, they will separate from one another. Release factor number 1, it recognizes two types of stop codons or termination codons, that is UAA and UAG. The release factor number 2, it recognizes UAA and UGA, whereas the third type of release factor, that is RF3, it dissociates the 30S and 50S subunit so that the machinery for synthesis of proteins is disassembled. This slide explains us the termination of translation. At the first point here in this particular image, we have a ribosome. The protein synthesis is being carried out on this ribosome. So here we have a tRNA molecule at the P site which has a polypeptide chain attached to it. So tRNA, which is present at the P site, it carries the completed polypeptide. Now at this A site there is a stop codon, either UAA, UGA or UAG. One of the three termination codons will be there now at the A site. So when this termination codon or the stop codon is there on the A site, that is within the reading frame, there is no tRNA molecule inside the cell which has an anticodon sequence complementary to this particular stop codon. So a tRNA molecule is not going to recognize this termination codon. Instead, a release factor will come and bind at the A site. Now there are three types of release factor depending upon the type of stop codon or termination codon at this position. A particular type of release factor will come and bind to the A site. So here we can see the release factor, it is bound at the A site. The polypeptide chain that is present attached to the tRNA at the P site gets released as soon as the release factor comes and attaches itself at this particular point. The tRNA is then released. So this is the finished polypeptide chain. It is released from the tRNA, and now the tRNA itself, which was now present at the P site, is also released from the ribosome. This is followed by dissociation of ribosomal subunits. The smaller and the larger subunits, they will get separated, as well as the template that is mRNA, which was being used for carrying out the translation, that is protein synthesis, it also gets separated. So in this manner, the termination of translation takes place inside bacteria. The newly synthesized proteins or polypeptides, they may not be biologically functional. They might undergo several foldings to create a secondary structure. They may undergo processing also, which is called as post-translation modification. Especially the amino terminal and carboxy terminal ends of the polypeptide chain, these are modified. In case of bacteria, the N-formylmethionine, that is the first amino acid in the polypeptide chain, it is removed from the polypeptide chain by enzymatic action. This results in formation of a functional protein or polypeptide.